Our aim was to assess the association between cognition, measured with general cognitive scores, and the gut microbiome structure. We used data from the third generation cohort from the Framingham Heart Study and filtered for middle-aged adults with no history of stroke or dementia and who had completed a cognitive assessment, as well as provided a stool sample. Some additional demographic characteristics were obtained and adjusted for, such as BMI, age, sex, education, and the time interval between the cognitive assessment and the stool collection. The stool sample was sequenced, and the microbiome composition was estimated using 16S RNA methods. And then finally, we use multivariable association and differential abundance analysis to estimate the association between the bacteria abundance and the general cognitive scores. Our general cognitive scores were computed based on neuropsychological assessment of 4 cognitive domains. Those included executive functioning, processing speed, language, and memory. And the composite score was used to estimate how cognitive dysfunction relates to the gut microbiome structure. In addition, participants were stratified into 2 groups. One was the poor cognition, and those were participants in the first quintile of scores, and then normal cognition was from the 2nd to the 5th quintile scores. So participants in the poor group had significantly increased abundance of a species bolteae, and through both multivariable association and differential abundance analysis, it suggested that poor cognition was associated with an increased abundance of trimethylamine producing species. And this has been linked to several detrimental metabolites, leading to cardiovascular disease, type 2 diabetes, obesity, suggesting that this could be a potential target for age-related diseases. And this species was only seen in the poor cognitive participants, leading us to believe that those who tested well didn't have this species. So it could be something either in their diet, their genetics, maybe medications that had led them to have an abundance of this bacteria. While these results are promising, there's still much to be analyzed, like I was saying, like with the diet, medication, genetics, and of course, like we know, cardiovascular disease, to look for some driving or mediating factors with the association of microbiome and cognition. So at this moment, we don't know what bacteria or maybe any other factors that could be driving or mediating, but that is something that we're going to be looking into in our next analysis, as well as more research needed for diverse populations. We did use the cohort from the Framingham Heart Study, which may not be reflective of the U.S. population. Since we do know, like I said, that this is a detrimental metabolite that has been seen in cardiovascular disease, maybe manipulating the abundance through either diet or pre or probiotics could be a research path for preserving global cognitive function. And we know that cardiovascular disease is a risk or is correlated with Alzheimer's disease. And our data is showing that this abundance of bacteria could also be a way to maybe have early detection or a part of the prognosis of Alzheimer's disease. I think that's something that we don't quite have a definitive answer for. This is it's an ongoing thing. This is just very preliminary data. But I think knowing that abundance is going to be a key role in it and not necessarily diversity. There's hundreds of thousands of different microbes and bacteria in the gut. So not necessarily the abundance, the diversity, but the abundance is going to play a key role. And, we never know yet if it's just a genetic thing, if it's maybe medications that affect this abundance, environmental factors, and of course, other comorbidities.